Good evening, all of you. Hi, Akash. So, good evening. This is Dr. Shanmugapriya, an academy educator for biochemistry. And this is session for NEET PG 2023 aspirants. And in this session, I've chosen few previous year questions. And using these previous year questions, I will be helping you understand few facts and concepts. And I've chosen latest NEET PG and INA CET questions for this purpose. Okay, so that's an introduction to this session. And before we start the session, let me tell you a few facts about an academy's plans. So what has been released recently is recorded sessions. So comprehensive series of every subject has been released as NEET PG Vital. So if you want to watch all these sessions at your own pace, at your own convenience, then you can take up the subscription, which is NEET PG Vital subscription. But that does not mean an academy has stopped live classes as well. There are also live sessions. So if you want to watch live sessions, you can take up NEET PG live subscription. If you want recorded sessions, NEET PG Vital subscription is what you want. And if you want a combo package, that's also available. Good evening, Dr. Bean. Good evening, Anialia. Hi. So we also have last leap marathon batch, which is for NEET PG 2023 aspirants, and the duration of this batch is 1 month. There are totally 1 or 2 hours of dedicated rapid revision session, which will be done by all educators. And the cost of this course would be just 3,199. And we also have FMG Mastery batch, which is an ultimate concise batch for FMG June 2023. Good evening, Lakshmi. Good evening, Yashwant. Hi, Yashwant. Yeah, good evening, Yashwant Saraswat. So, hi, all of you. Happy to meet you all. And because we are celebrating Republic Day, we have up to 20% subscription plan, and this is valid till January 28. Okay, so that's all about an academy's plans. Give me a minute. So this is the first question for all of you. This is a NEET PG 2021 question, and I hope most of you must have seen this MCQ, right? And most of you would have found it difficult to understand this question. So the question is actually a very simple question. How many questions? I think I have around 15 MCQs. It will be useful. Okay? Yeah. So you're conducting an experiment on mitochondrial. Good evening, Meghavat. Do you plan of entire biochemistry sessions again in an academy app? I won't be able to finish entire biochemistry series. If you're asking me as a special class, no, I don't think I'll be able to do it. As plus class, there will be comprehensive courses which are coming up every now and then. Yeah, that's possible. Very good, Bhavesh. Bhavesh has answered it as oligomycin. So, thank you, Karishma Singh. Good evening, Avinash. Yeah, answer is oligomycin. So let me read the question for all of you so that I'll be able to comprehend the question for you. There is somebody who's doing an experiment on mitochondrial respiration, and the person has added malate, pyruvate and succinate, and the respiration is normal. But when a new substance was added along with the above mentioned substrates, the respiration is blocked. I want you to understand the difference between respiration and phosphorylation. What do you mean by respiration? Respiration is nothing but oxidation of fuels. So what is affected? They are telling you that oxidation of fuel is affected. Okay, which means indirectly phosphorylation also will get affected. Okay, which of the following substances is most likely added? What should be our exclusion? The first choice that you should exclude is 2,4-dinitrophenol. That definitely is not the answer here. Because what is 2,4-dinitrophenol? It is an uncoupler. Now, what is electron transport chain? Electron transport chain couples oxidation with phosphorylation. So, if you ask what does electron transport chain do, your answer should be it couples oxidation of fuels with phosphorylation of ADP to form ATP. Now, what does an uncoupler do? Uncoupler uncouples oxidation from phosphorylation, or in other words, in the presence of an uncoupler, oxidation of fuel will happen continuously without phosphorylation. So in the presence of uncoupler, the only process that gets affected is phosphorylation, but what happens continuously? Oxidation of fuel happens continuously, and continuously energy is getting liberated, and that energy will be liberated out as heat. And that is why we always say that uncouplers cause heat production. Okay, so do you think uncoupler can be an answer here? Because they've told you that respiration is blocked. It is definitely not an uncoupler, because in the presence of an uncoupler, respiration is not blocked, only phosphorylation is blocked. Do you all understand this? Yeah. Now about these three choices. To understand these three choices, you will have to know about complexes of electron transport chain. At what level do these inhibitors act? So I'm going to tell you all about that now. Listen to this. At the end of the session or at the end of this MCQ discussion, you'll be able to clearly talk about electron transport chain and its role in ATP production. How do these inhibitors act? That's my assurance to you. Okay. So what are the electron transport chain complexes? You know, these electron transport chain complexes are arranged along the inner side of inner mitochondrial membrane. There are 5 stationary complexes and 2 mobile complexes. Use bold pen and highlighters for important points. Yes. Yes, Pickle, that's what I'm doing. Yeah, I will do it. So there are 5 stationary complexes. Thanks for all the suggestions. I will do it. So there are 5 stationary complexes and 2 mobile complexes. Those 5 stationary complexes will be numbered in Roman letter, complex 1 till complex 5. Now what is complex 1? Complex 1 is NADH linked dehydrogenase. That is, electron from NADH enters into electron transport chain through complex 1. Okay. Now what is complex 2? Complex 2 is succinate dehydrogenase. Do you remember in the question, one of the substrates which was added was succinate. So complex 2 is succinate dehydrogenase. Complex 3, yes, very good, Yashwant. Complex 3 is cytochrome B and C1. Complex 3 is cytochrome B and C1. Complex 4 is cytochrome A and A3. And complex 5 is ATP synthase complex. Is that clear? These are the 5 stationary complexes. And between these 5 stationary complexes, you have mobile complexes Q and C. Okay. So ubiquinone or Q is placed between complex 1 and complex 3. Ubiquinone is placed between complex 1 and complex 3. Cytochrome C is placed between 3 and 4. Okay. This is how they are arranged. Now, what is the direction of electron here? If the electron comes from NADH, it enters into electron transport chain through complex 1. So, if it is pyruvate dehydrogenase or malate dehydrogenase, which gives rise to NADH, that will enter into electron transport chain through complex 1. From complex 1, electrons enter into Q. From Q, electrons go to 3. From 3, electrons go to C. From C to 4. Can you all tell me what is the final acceptor of electron? Quickly answer in the live chat, please. What will be the final acceptor of electron? It is oxygen. Okay. So this is if it is NADH. If it's FADH2, it enters into electron transport chain through complex 2. From complex 2, very good Dr. Sarika, very good Dr. Ruhi. From complex 2, electrons go to Q. Yeah, from here you know Q to 3, 3 to C, C to 4 and 4 to oxygen. So this is the direction of transport of electron. Okay. And I want you to remember that as and when electrons get transported across these electron transport chain complexes, these complexes are arranged in the order of decreasing energy level. How are they arranged? Decreasing energy level. So when electrons move from one complex to another, energy will be liberated and that energy is used for pumping hydrogen ions. For now, just remember that. When electrons go through these electron transport chain complexes, because electrons move from a complex of higher energy to a complex of lower energy, energy is liberated that is used for pumping what? Hydrogen ions. Okay. Now, where do these electron transport chain inhibitors act? I want the chat box to be active. Yes, you're right, Yashwant. Very good. So, electron transport between complex 1 and Q here. Yeah, electron transport between 1 and Q is inhibited by all the names start with A. What is the first alphabet, all of you? The first alphabet is A. Between 1 and Q is inhibited by number 1, amobarbital. Yeah, please tell me what is it? It is amobarbital. Pyrisidin A. Yeah, pyrisidin A ends with A. And then rotenone. Yeah, rotenone is not vitamin A, but it sounds like vitamin A, right? Hi, AC. Okay, so tell me what are the inhibitors which inhibit electron transport between 1 and Q? All these three names have got something to do with A. Amobarbital starts with A, pyrisidin A ends with A, and rotenone sounds like vitamin A. Okay, now complex 2 or succinate dehydrogenase. Succinate dehydrogenase is inhibited by malonate. Now electron transport between 2 and Q, between 2 and Q. Here it is inhibited by TTFA. It is inhibited by TTFA. How to remember it? 2 starts with T, right? It's inhibited by TTFA and carboxyl. Electron transport between 3 and C is inhibited by something with 3 letters. What has got 3 letters? BAL. Complex 3 and cytochrome C is inhibited by BAL and antimycin. So please remember, this is antimycin. It starts with A. It has got nothing to do with complex 1. So BAL and antimycin inhibit electron transport between complex 3 and cytochrome C. And electron transport between complex 4 and oxygen, the final acceptor between 4 and oxygen, is inhibited by common poisons. What are the common poisons? Hydrogen sulfide, cyanide and carbon monoxide. Very good. Hydrogen sulfide, cyanide and carbon monoxide. Such an oligomycin does not act here. Oligomycin inhibits ATP synthase complex, about which I will tell you at the last. Okay. So hydrogen sulfide, cyanide and carbon monoxide inhibit electron transport between complex 4 and oxygen. So this is how inhibitors act. Okay, now let's look at the choices here. Have a look at these choices. We have already excluded 2,4-dinitrophenol, and among these choices I am going to exclude rotenone. Can anybody tell me why am I excluding rotenone? Because rotenone acts where? Rotenone acts here. If it was rotenone, it should have inhibited respiration only after adding malate and pyruvate. Do you understand this? If it was rotenone, it should have inhibited respiration after adding malate and pyruvate. What should have not been inhibited? Succinate oxidation should have been not inhibited. It should have happened normally. The respiration should have happened normally. But that's not what has happened here. So this is not rotenone. So I have excluded rotenone. And what am I left with? I am left with antimycin and oligomycin. Where does antimycin act? Antimycin inhibits electron transport between complex 3 and cytochrome C. That is a possibility, because if you are inhibiting here, both malate pyruvate and succinate oxidation will definitely be inhibited. Okay, so this is a possibility. But what is also a possibility? It is also a possibility that it is oligomycin. How does oligomycin act? As I told you, when electrons go through these electron transport chain complexes, where are these complexes arranged? They are arranged along the inner side of inner mitochondrial membrane. When electrons go through these electron transport chain complexes, what is translocated? I told you, hydrogen ions get translocated. From where do they get translocated? From the mitochondrial matrix to just outside the inner mitochondrial membrane. Yes. Hydrogen ions usually get translocated from the matrix to just outside the inner mitochondrial membrane. After these hydrogen ions accumulate outside the inner mitochondrial membrane, these hydrogen ions find their way through FO component of ATP synthase complex. So what is complex 5? Complex 5 is ATP synthase complex, which has got FO and F1 subunit. What is FO subunit? It's an ion of 4 through which hydrogen ions can go. And what is F1? F1 is the ATP synthase enzyme. And here hydrogen ions move from a region of higher concentration to a region of lower concentration. Yes or no? Here hydrogen ions move from a region of higher concentration to a region of lower concentration. That liberates energy. That energy is used for phosphorylating ADP to form ATP. So this is how electron transport chain works. Now how does oligomycin act? Oligomycin acts by inhibiting FO component of ATP synthase complex. That is why it is FO. Why is it FO? To denote that it is inhibited by oligomycin. So if it comes and inhibits here, can you tell me what are the steps that are prohibited? Hydrogen ions cannot go through FO component, so ATP cannot be synthesized. So on one hand, it inhibits phosphorylation. On the other hand, because hydrogen ions are not able to go through FO component, in due course, because there is no hydrogen ion, respiration also gets affected. So which do you think will be a better answer, all of you? Which do you think will be a better answer between these two choices? I am going for oligomycin because oligomycin goes and inhibits the ultimate step. The ultimate step wherein ATP can be produced. Once that is inhibited, there is no hydrogen ion movement. Without hydrogen ion movement, electron transport and respiration also gets affected. And that is why what is the right answer, all of you? That is why the right answer is oligomycin. So have you understood the logic behind how we can exclude all these choices? Yeah, 2,4-dinitrophenol cannot be considered because it does not inhibit respiration. Rotenone cannot be considered because if it was rotenone, it should have inhibited only malate and pyruvate oxidation. It should not have inhibited succinate oxidation. Okay, because it is inhibiting after that, I am excluding rotenone. Between antimycin and oligomycin, I am preferably choosing oligomycin because it inhibits the ultimate point. Okay, so that's all about this choice. Highlighters. Why are you so bent on highlighters? Okay, I will use that pickle. A child with loose stools for 1 day presents with dehydration. So the point here is dehydration. She is prescribed oral rehydration solution. Sodium present in oral rehydration solution is absorbed through which of the following apical site transporters? Many of you have answered it as choice B. Lakshmi Kumari, very good. Lakshmi Kumari has answered it as choice A. Yes, what is the right answer here? The right answer here is sodium glucose cotransporter 1. Okay, answer is sodium glucose cotransporter 1. Let's it will make your clinical more interesting. Sure, people, I will do that. So let's try to exclude choices one by one. Let me first help you exclude choice C and choice D. Yeah, we are trying to understand about GLUT4 and GLUT2 transporters. So I want your help in filling up this tabular column. So first tell me GLUT1 and GLUT2 transporters belong to which kind of transport mechanism? All GLUT transporters belong to facilitated passive diffusion mechanism. Is that clear? All GLUT transporters belong to facilitated passive diffusion mechanism. Hang on for some more time. I am going to tell you about all transport mechanisms as well. For now, just remember it is facilitated passive diffusion. Okay. So tell me, where do you find GLUT1 and GLUT3 transporters? Both GLUT1 and GLUT3 transporters are present in those cells for which glucose is indispensable. So what are the cells for which glucose is indispensable? It is indispensable for RBCs, neurons and placenta. Okay, so GLUT1 and GLUT3 transporters are present in RBCs, neurons and placenta. Now how about GLUT2 transporters? GLUT2 transporters are present in those cells for which are related to glucose balance. So tell me, what are the cells which are related to glucose balance? First, glucose has to be absorbed by enterocytes, and then glucose has to be stored by hepatocytes. Pancreatic beta cells should release insulin, and glucose has to be reabsorbed by proximal convoluted tubular cells. So tell me, what are the cells which are related to glucose balance? Enterocytes, hepatocytes, pancreatic beta cells and renal tubular cells. And in all these cells, which transporter is present? It is GLUT2 transporter that is present. Okay. Now about GLUT4 transporters. Who wants to answer about GLUT4 transporters? These GLUT4 transporters are favorite questions of both NEET PG, INI, CET and FMG. So GLUT4 transporters, insulin dependent glucose uptake. Okay, insulin dependent glucose uptake happens where? It happens in only those tissues wherein glucose can be used for anabolism. Where can you use glucose for anabolism? Skeletal muscle, cardiac muscle and adipose tissue. How can you use glucose for anabolism in skeletal muscle and cardiac muscle? Because both in skeletal muscle and cardiac muscle, glucose will be stored as glycogen. In adipose tissue, glucose will be stored as fat. And in all these places, insulin dependent glucose uptake happens in this question. 2 options were right. Is that right? Is that true? So skeletal muscle, cardiac muscle and adipose tissue, what is the transporter that you have? You have GLUT4 transporter. Okay. Now GLUT5 transporter is related to fructose absorption. GLUT5 is related to fructose absorption. So have you understood all about these GLUT transporters? So this is the tabular column that I wrote just now. Yeah. So after the session, you can download the PDF and use this to remember all these facts and concepts. So it's all about repetition. All of you, take it from me. It's all about repetition. Today you might think you have memorized everything, but tomorrow you're going to forget it. So to avoid this, please repeat it as many times as possible. Okay. So GLUT1 and GLUT3 transporters are present in neurons, RBCs and placenta. GLUT2 in enterocytes, pancreatic beta cells, hepatocytes and in proximal convoluted tubular cells. GLUT4, everybody should remember. Anytime you're asked, you should be able to answer this. Insulin dependent glucose uptake happens in skeletal muscle, cardiac muscle and adipose tissue by GLUT4 transporter upregulation. F for F. GLUT5 transporters are related to fructose absorption. Okay. So which transporter did I tell you is present in enterocyte? Tell me. I said enterocytes have which transporter? GLUT2 transporter. Now, where do you find this GLUT2 transporter? Are these GLUT2 transporters present along the apical side or along the basolateral side? Your GLUT2 transporters are, if asked to choose one, then in GLUT4 transporter, adipose tissue should be marked. What was the other choice? Adipose tissue and muscle. If you find adipose tissue, go for it, pickle. Okay. So where do you find this GLUT2 transporter? GLUT2 transporters are present along the basolateral side. What is present along the apical side? Along the apical side, you have sodium glucose cotransporter 1. All of you please memorize it. Along the apical side, you have sodium glucose cotransporter 1. Along the basolateral side, you have GLUT2 transporters. And only because, at last, please help us to tell how to grab high yield revision in short time. Yes, Dr. Bean, I will do that. I'll definitely do that. Okay. So along the apical side, you have sodium glucose cotransporter 1, and that is why in oral rehydration solution, you do not only add sodium. What is the purpose of you giving oral rehydration solution to any person? It is to replenish the sodium loss, for which do you add only salt? No, you add salt and sugar. What is the purpose of you adding sugar? What is table sugar, all of you? Table sugar is sucrose. When you ingest sucrose, this sucrose gets digested to give glucose and fructose. And whenever glucose gets absorbed, along with that, what also gets absorbed? Sodium also gets absorbed. And that is why, to facilitate sodium absorption, you add salt and sugar in oral rehydration solution. If you remember that, you will know that along the apical side, what is that we have? Along the apical side, you have sodium glucose cotransporter 1, and along the basolateral side, you have GLUT2 transporters. Depending on you for biochem, yes, I am dependable as far as biochem is concerned. Don't worry, Sriram. Please watch all these sessions. Yeah, you'll be able to answer all the MCQs. Okay. So GLUT2 transporters, they belong to which type of transport mechanism? I told you they belong to facilitated passive diffusion mechanism, whereas sodium glucose cotransporter 1 will be secondary active transport. All of you please memorize it. Whenever I call something as a co-transporter or counter-transporter of sodium, you can close your eyes and say they belong to secondary active transport. Except sodium, potassium, ATP is formed, everything else will be secondary active transport that is present along the apical side. So now tell me the answer for this question. Sodium present in oral rehydration solution is absorbed through which of the following apical side transporter? What will be our answer? It is sodium glucose cotransporter 1. Where do you find sodium glucose cotransporter 2? Cotransporter 2 is present in proximal convoluted tubules. GLUT4 under the influence of insulin. Yes, dark night. That's what we discussed previously. Okay. So this is the next question. Again, INI-CET 2021 question. What is the metabolic fuel for RBCs during starvation? All of you tell me the answer. Which metabolic fuel is used by RBCs during starvation? It is glucose. Somebody has answered it as ketone bodies. Dr. Reformer, it is not ketone bodies, Reformer. What does it use? It always uses glucose. Why? Can it use ketone bodies? How do you use ketone bodies? You use ketone bodies through citric acid cycle. For citric acid cycle to happen, what is a suborganel that you want? Mitochondria. Does your RBC have mitochondria, all of you? No. So will you ever say RBCs use ketone bodies? Please don't say that. Please remember, RBCs do not have mitochondria. They can never utilize ketone bodies. Got it? So what is the right answer here? The right answer here is glucose. Okay. So whenever they ask you any question on choice of fuel, the first question that you should ask to yourself, the first question that you should ask to yourself is it aerobic or anaerobic cell? What should you find out? You should find out whether the cell is aerobic or anaerobic. If it's anaerobic, you can close your eyes and say the cell is going to use glucose, because the only pathway that can generate ATP anaerobically. I will tell you about that, Dr. Bean. Listen to this. All anaerobic cells use what as their preferred fuel? They use glucose as their preferred fuel, because the only fuel that can be used anaerobically is glucose, because the only pathway that can generate ATP anaerobically is anaerobic glycolysis. Don't we know that? Anaerobic glycolysis gives rise to 2 ATPs. So what are the examples of anaerobic cells? RBCs, which do not have mitochondria, right? RBCs, retinal cells, corneal cells. And then white muscle fibers. Who wants to tell me why are red muscle fibers red? Please tell me, why are red muscle fibers red? Red muscle fibers are red because they have myoglobin, which is a storage form of oxygen. So red muscle fiber with myoglobin, which is a storage form of oxygen, will undergo aerobic metabolism. Whereas white muscle fibers which do not have myoglobin will have to undergo anaerobic metabolism. When they undergo anaerobic metabolism, what is the fuel white muscle fibers will use? All white muscle fibers use glucose as their preferred fuel. Got it? And apart from white muscle fibers, renal medulla. Do you all know that renal medulla receives very less blood supply? Renal medulla receives very less blood supply because you must have learned that renal medulla receives blood supply through end arteries. Yeah, that is why whenever there is an ischemic damage to kidney, it's the medulla which gets affected the most. Okay, so because it receives less blood supply, that also preferably uses glucose as its preferred fuel. Okay, yes, corneal cells also. Okay, so now about aerobic cell. If you get to know that the cell is aerobic, you can say the cell is going to use fatty acids, because though aerobic cell can use both glucose and fatty acid aerobically, given a choice, what will a cell choose? The cell will choose fatty acids, because fatty acids on oxidation will give rise to more number of ATPs than glucose on oxidation. Glucose on complete oxidation gives rise to how many ATPs, all of you? Glucose on complete oxidation will give rise to only 32 ATPs. Whereas fatty acids on complete oxidation, for example, palmitic acid on complete oxidation. Whoever is watching this session, any fact which I tell you, please memorize it every now and then. Okay, don't think I'll watch the session later on, I'll memorize facts. That never happens. Okay, so whenever I tell you a fact, please memorize it. So from now on, if somebody asks you glucose on complete oxidation gives rise to how many ATPs, what should be your answer? 32 ATPs. Palmitic acid on complete oxidation gives rise to, very good, Dark Knight. Yes, it is 106, Dr. Bean. It is 106 ATP. So if I give you a choice, what will you choose? You have to choose fatty acids. So all aerobic cells use fatty acid as their preferred fuel. And that is why cardiac muscle fibers and red muscle fibers use fatty acid as their preferred fuel. Do you understand it now? Cardiac muscle fibers are known for their mitochondrial content. No worries, Dr. Bean. You're watching this session only to revise and recollect. Yeah. Till now, if you get confused, it's not a problem. Now you can memorize it. So cardiac muscle fibers are known for their mitochondrial content. They use aerobic oxidation, so they use fatty acids as their preferred fuel. Red muscle fibers also preferably use fatty acids as their preferred fuel. So fatty acids will be stored in red muscle fibers as triacylglycerol and cholesterol ester. And that is why you're supposed to refrain from having red meat. Have you heard of it? If somebody comes to you with hypertriglyceridemia and hypercholesterolemia, and if the person asks you what should I do as a dietary change, what as a medico you would say? You would say refrain from having red meat. Why do you say that? Because red meat, red muscle fibers preferably use fatty acids. So they store fatty acids as triacylglycerol and cholesterol ester. So anybody who consumes red meat will end up getting hypertriglyceridemia and hypercholesterolemia. Got it? Yeah. So that's about red muscle fibers and cardiac muscle fibers. And there is one exception to this rule. Whereas one aerobic cell uses glucose as its preferred fuel. Which aerobic cell uses glucose as its preferred fuel? Type 1 is red pH. Type 1 is red oxidative slow twitch. Can you memorize it that way, pH? For type 1, you can have a mnemonic which is ROS. Have you heard of reactive oxygen species? So type 1 is red muscle fiber. It is oxidative muscle fiber. It is a slow twitch muscle fiber. Got it? Yes. Very good Minu. Very good Dr. Ruhi. Very good Divya. Yes. There is one aerobic cell which uses glucose as its preferred fuel. Why are white muscle fibers fatigable? Okay, I will tell you the difference between red muscle fiber and white muscle fiber now, Guru. Okay, after discussing this MCQ, I will tell you. Okay. So, what was I telling you? Yeah. So, neurons use glucose though they are aerobic. Why do you think neurons use glucose though they are aerobic? Because your neurons, most of your neurons are surrounded by what? Blood-brain barrier. And as long as they are surrounded by blood-brain barrier, what cannot cross it? Your fatty acids in the circulation will be attached to albumin. And anything which is attached to albumin cannot cross the blood-brain barrier. Because your neurons do not have an access to fatty acid, your neurons have evolved to use only glucose as their preferred fuel. So neurons use glucose aerobically. RBCs use glucose anaerobically. Got it? Yes. So that's about preferred fuel. So can we fill up this tabular column? Yeah, I'm going to tell you what will be the preferred fuel for all the cells in the fed state, in fasting state and in starvation. Okay, you're going to help me fill it up. So let's start with RBCs. Why am I starting with RBCs? Because it's very easy. RBCs do not have mitochondria. So do you have any choice? We don't have any choice other than using glucose. So RBCs use glucose irrespective of whether it is fed state or fasting state or starvation. Got it? Similarly, neurons. How about neurons? Neurons also preferably use glucose. They use glucose in the fed state and even in the fasting state. And that is why in fasting state, your primary aim is to maintain blood glucose. Do you understand this? In extremes of starvation, they switch over from using glucose to using ketone bodies. Do you understand this? When in extremes of starvation, they start using ketone bodies. So that's about RBCs and neurons, which are straightforward. Now about red muscle fibers. You will have to tell me the answer. Red muscle fibers, they are aerobic. So what will be their preferred fuel? Aerobic cells use fatty acid. So red muscle fibers use fatty acid both in the fed state and in the fasting state. In starvation, they start using ketone bodies. Okay, that's about red muscle fibers. And the same is applicable to cardiac muscle fibers. What is that you know about cardiac muscle fibers? Their preferred fuel will be fatty acid in fed state and fasting state. In starvation, they will start using ketone bodies. Now tell me about white muscle fibers, all of you. White muscle fibers, they don't have myoglobin. So they don't have a storage form of oxygen. In that case, what will happen when your white muscle fibers contract? When your white muscle fibers contract, blood vessels get constricted. So they don't get a fresh supply of oxygen. They don't have a storage form of oxygen as well. So they become dependent on what? Anaerobic metabolism. So white muscle fibers preferably use what? Glucose. In the fed state and in fasting state, they use glucose. Yeah. And in extremes of starvation, they also start utilizing ketone bodies. Okay. That's about white muscle fibers. Now tell me about liver. Liver is totally different. I'm giving you 10 seconds time to think about liver. What do you think liver uses in the fed state? Does liver use glucose or does liver use fatty acid in the fed state? Tell me. In the fed state, liver neither uses glucose nor uses fatty acid. Liver uses amino acids. Why do you think it does not use glucose or fatty acid? Because liver has got which enzyme? Yeah, never ketone body. Very good, Gaurav. Excellent. Yeah, liver. Why do you think liver does not use glucose? Because liver has got an enzyme glucokinase. What is the enzyme that is present in liver? Glucokinase. And this glucokinase has got very low affinity for glucose. And nature has purposely given your liver glucokinase with very low affinity for glucose. Otherwise, liver would have used all glucose for its own. Any dietary glucose reaches liver first. If liver was provided with an enzyme with very high affinity for glucose, liver would have used all glucose for its own, so that peripheral tissues suffer from hypoglycemia, which should not happen, right? That is why we are provided with glucokinase in our liver, which has got very low affinity. So even in the fed state, liver does not use glucose as its fuel. Why do you think liver does not use fatty acid in the fed state? Because any dietary lipid gets absorbed as chylomicron. And this chylomicron gets into lacteals, right? They get into lymphatics. Through lymphatics, they get into thoracic duct. Through thoracic duct, they reach the extrahepatic tissues first. They don't reach liver. So what reaches liver, which liver can use? Liver uses amino acids. Is that clear? Can you tell the pathway for energy in various states? That's what we are discussing, Dr. Reformer. That's what we are discussing, pathways. Okay, I will tell you about what happens in the fed state and what happens in starvation in the next YouTube session. Okay, so after this I will tell you about differences between type 1 and type 2 muscle fibers. For now, what is the preferred fuel that is used by liver? Liver uses amino acids. Okay, so even in fasting state, once it gets into fasting state, it starts using fatty acid. Because in fasting state, adipose tissue peripheral lipolysis happens. Yeah, in adipose tissue, peripheral lipolysis happens, that starts pushing in fatty acid. So liver will start using fatty acid. And in starvation also it uses fatty acid. Have you understood this tabular column? Yeah. So again, you can download this PDF, you can sit, read and understand and memorize what is the preferred fuel in various states. Okay. So before we start, so tell me the answer for this. What is a metabolic fuel for RBCs during starvation, all of you? RBCs during starvation, be it the fed state or starvation, what does it use? It uses only glucose. Okay, and because I think someone was asking me about the differences between type 1 and type 2 muscle fibers. Can we discuss that now? Yeah, type 1 and type 2 muscle fibers. Type 1 muscle fibers are red muscle fibers. Type 2 muscle fibers are white muscle fibers. Okay. So what does that mean? It means type 1 muscle fibers have myoglobin. Type 2 muscle fibers do not have myoglobin. Okay. Now what if type 1 has got myoglobin? It undergoes what metabolism? Aerobic metabolism. Because it undergoes aerobic metabolism, I will call it as an oxidative muscle fiber. Now type 2, which does not have myoglobin, will have to undergo anaerobic metabolism. And what is the only pathway which can generate ATP anaerobically? It is glycolysis. So type 2 muscle fiber will be otherwise called as glycolytic muscle fiber. Do you understand this? Because type 2 uses glycolysis, you will call type 2 muscle fiber otherwise as glycolytic muscle fiber. So tell me, what will be the preferred fuel for all these cells? Because type 1 is oxidative, it uses fatty acid as its preferred fuel. Type 2 will use glucose as its preferred fuel. So fatty acid here will be stored as triacyl glycerol and cholesterol ester. Here glucose will be stored as glycogen. Okay. Now type 2, which is glycolytic muscle fiber, the rate of onset of ATP production will be faster. Because as far as glycolysis is concerned, it is substrate level phosphorylation steps. And all these substrate level phosphorylation steps, you get ATP directly. Whereas if it is fatty acid oxidation, you don't get ATP directly. Do you remember in every fatty acid oxidation pathway, all that you can expect is NADH or FADH2. And then this electron transport chain should convert NADH and FADH2 to ATP. That will take some time. Okay, so in fatty acid oxidation, the rate of onset of ATP production will be slower. Whereas in type 2 or glycolytic muscle fiber, because it is substrate level phosphorylation, the rate of onset of ATP production will be faster. So when will you use type 2 muscle fiber? For fast twitchers, wherein the onset has to be faster. Whereas type 1 will be used for slow onset contraction. That is why type 1 is called as slow twitch muscle fiber. Type 2 will be called as fast twitch muscle fiber. Have you all understood this? Though the onset of ATP production is faster in type 2, because it is glycolysis, do you think it can go on forever? No. When it is anaerobic glycolysis, in due course, anaerobic glycolysis gives rise to lactic acid. That lactic acid suppresses glycolysis in due course. So it cannot be a sustained muscle. Do you understand this? This muscle cannot bring about a sustained contraction. So when will you use type 2 muscle fiber? For those contractions which need faster onset, which need not be continued for a prolonged period of time. Because in due course, ATP production will stop because glycolysis is terminated. So it is used for faster twitches but cannot be prolonged. Do you now understand this? I think Guru was asking this question, right? Guru, have you understood it? It can't be prolonged for a longer period because it gets inhibited. Anaerobic glycolysis gets inhibited. Now type 1, though the onset is slower, it can be sustained. So type 1 muscle fiber will be used for slow and sustained contraction. Type 2 muscle fiber will be used for faster contractions which need not be continued for a prolonged period. And as someone has answered it, Van has answered it, that is why type 2 will be used by sprinters and type 1 will be used by marathon runners. Okay, so this is about the concept. Must know. All these are must know facts. So if you ask me what are the must know facts here, for type 1 it is red, it is oxidative, it is slow twitch. Do you understand this, Pickle? Yeah, type 1 is red, oxidative, slow twitch. That is why I gave you a mnemonic. What is a mnemonic I gave you for type 1? It is reactive oxygen species. I am happy that most of you have understood it. So what is type 1? It is reactive oxygen species, ROS. We should not sprint, ma'am, go slow, we get fatigued. Yes, Pickle. Yes. Are you PK? Is Pickle the same as PK? Okay, so what is the right answer here? The right answer here is glucose. If it's PK, I remember PK saying the last time, yes, the last time he was, hi Mohammed Guljar, I'm happy. Yeah, so this is again an INI CET 2021 question. This topic I've never discussed, so I thought I should discuss it today. Proteins can be precipitated by all except. What do you think is the right answer? Adding trichloroacetic acid, moving pH away from isoelectric pH, adding acetone, adding sulfosalicylic acid. What do you think is the right answer? The right answer is moving pH, moving away. Very good, Guru. Moving away from isoelectric pH will not bring about protein precipitation because at isoelectric pH proteins get precipitated. Okay, so let us discuss how to precipitate proteins. Last time in the sense the last NEET PG when you were preparing, Pickle used to say that he used to watch all sessions in 2x speed. Okay, so I would like to reiterate this to everybody. You cannot be in a hurry. Yeah, you cannot be in a hurry for something which you want to be long lasting. For example, memory has to be long lasting, and if you want to memorize something, you should not be in a hurry. Spend enough time, try to understand concepts. Enough time should be spent in memorizing all the facts. No point just hurrying and not memorizing anything. That's what I was trying to say. Okay, no worries. No worries, people. You can do it this time. Okay, so why do you think this will not help in protein precipitation? I will tell you about principles of protein solubility. When do you think a solute will be soluble in a solvent? Only when a solute can interact with a solvent. When will a solute be able to react or interact with a polar solvent? Only when it is charged. So there are two factors which allow a protein to be soluble in a polar solvent. One is the charged shell. Okay, one is the charged shell. The other one is a hydration shell. So tell me, what is responsible for solubility of a protein in a solvent? It is two shells. One is the charged shell, the other one is a hydration shell. So if you want to precipitate a protein, you should remove both these shells. That is your aim. Only then you can precipitate a protein. So how can you remove the charged shell? By bringing the protein to isoelectric pH. Because what is isoelectric pH? At isoelectric pH, the net charge carried by the particle is 0. When the charge is 0, there is no charge shell. When there is no charge shell, it cannot attract a hydration shell. So it will not be able to interact with the polar solvent and the protein will get precipitated. That is why at isoelectric pH, the solubility of the protein will be the least and the protein will get precipitated. And that is what we try by adding acids like sulfosalicylic acid or trichloroacetic acid. So when you add sulfosalicylic acid or trichloroacetic acid, you add acids in such a way that these proteins reach their isoelectric pH. Once they reach their isoelectric pH, they no more have the charge shell, they get precipitated. Okay. Another way of precipitating the protein is by removing the hydration shell. How can you remove the hydration shell? By adding a non-polar solvent like acetone. Acetone removes the hydration shell, will cause precipitation of proteins. Do you all understand this? Yeah. So now tell me the right answer. Proteins can be precipitated by all except. You can precipitate it by adding any acid and bringing the protein to isoelectric pH like trichloroacetic acid or sulfosalicylic acid. By bringing the protein to isoelectric pH, not away from the isoelectric pH, by adding an organic solvent which will remove the hydration shell. Okay? Yes. Hayes test. No pickle. Hayes test is done to detect bile salt. Yeah, what is the function of Hayes test? It is done to detect bile salts. And what is the principle used here? Bile salts reduce the surface tension. It reduces the surface tension of urine. So how do you perform Hayes test? You take a test tube where you add the urine which is suspected to have bile salts. If there is bile salt, it reduces the surface tension. So that when you add sulfur powder, what color is sulfur powder? Sulfur powder is yellow in color. So when you add sulfur powder, when this urine does not have surface tension, sulfur powder will sink to the bottom. Do you understand this? Sulfur powder will sink to the bottom. So that is Hayes test principle which is used to detect bile salts. Sulfosalicylic acid, yes Pickle, you are right. Sulfosalicylic acid is used to precipitate protein and to find out if there is protein in urine. Got it? Sulfosalicylic acid test, Pickle. Okay. So this is another important question. miRNA is used for what? So miRNA or siRNA, irrespective of however the question is asked. In INI CT 2021, there were two questions on miRNA and siRNA. What is it? Gene knockdown. Very good. Yes. So there were two questions on this. So miRNA or siRNA, what do? They help in gene knockdown. Excellent. Pickle is answering it as gene knockout. It is not gene knockout. What is it? It is gene knockdown. Okay. So first let me tell you about all these terms. I will first define all these terms and then I am going to let you answer this question. So what is gene knockdown? Gene knockdown is nothing but down regulating the gene expression. What do you mean by down regulating the gene expression? In this case, you don't touch the gene. You are not changing the gene sequence. Yeah, you are not changing the gene sequence. What do you mean by gene expression? You know, these genes on transcription give rise to mRNAs. Right? These genes on transcription give rise to mRNAs. These mRNAs give rise to proteins. Now, depending upon the number of times of transcription of the gene, number of mRNAs that you get will differ. Depending upon that, number of proteins that you get will differ. Now, what do you mean by down-regulating gene expression? By any mechanism, if you can decrease the number of times of transcription of a gene, decreasing the number of mRNAs produced, decreasing the number of proteins produced, that is called as down-regulation. In this case, you don't touch the gene sequence. Got it? Now, what is gene knockout? Can you please tell us the difference between siRNA? I am telling you, Krishna Patil, can you please not spam everybody? Yeah, so that's about gene knockdown. Now about gene knockout and gene knock-in. Gene knockout is nothing but you removing a defective gene. What is gene knockout? It is removal of the defective gene. What is gene knock-in? It is replacing a defective gene with a normal gene. Do you understand this? What is gene knock-in? It is nothing but replacement of a defective gene with a normal gene. In these two cases, whenever I say I am going to remove the defective gene or I am going to replace the defective gene with a normal gene, it is like me editing the gene. Yeah, I am editing the gene sequences. That is never done by your miRNA or by siRNA. Do you understand this? This is never done by miRNA or by siRNA. So what exactly do they do? So these miRNAs or siRNAs, the basic difference between miRNA and siRNAs, miRNAs are mostly endogenous. siRNAs can be endogenous or exogenous, but ultimately the function would be the same. Okay? So I'm going to tell you about what do these miRNAs do. So all these miRNAs and siRNAs have their own sequences. And suppose these siRNAs find complementary sequences. Suppose these siRNAs, I can't block anybody here, I don't have that ability. Hi Ankush Sharma, the Wolverine. Yes. So suppose these siRNAs find complementary sequences on any of the fully formed mRNAs. These siRNAs go and hybridize with those mRNAs and they form a double-stranded RNA structure. Not knowing that this double-stranded RNA has been formed, ribosome will start translating these mRNAs. You know the function of ribosome is to translate these mRNAs. So when these ribosomes start translating these mRNAs, they start moving ahead, and when they come across this double-stranded RNA structure, they know that it is not normal. During translation, finding a double-stranded RNA is abnormal. So what will these ribosomes do? These ribosomes recruit RNA transcription silencing complex. What do you call it as? You call it as RNA transcription silencing complex. And one of the proteins that is present in this RNA transcription silencing complex is an endonuclease. And this endonuclease makes multiple nicks on the mRNA. So what happens to this mRNA? mRNA gets fragmented. And once mRNA gets fragmented, there is no translation and the protein is not synthesized. So do you understand that you are down-regulating the gene expression, all of you? Do you understand that you are down-regulating the gene expression? You are not allowing. Please don't mind. Yeah, please don't mind anybody who's trying to spam. You just watch the session, okay? You just continue watching the session because it's an important fact or an important concept that everybody should know, okay? So what are we doing here? We are trying to down-regulate the gene expression because what is happening? The gene is forming the RNA, but mRNA is not allowed to be translated to form a protein. So what are we doing here? We are down-regulating the gene expression. So will that cause a disease? It is not going to cause a disease. It is going to be a way by which your gene gets regulated. This is one of the mechanisms by which we regulate gene expression. Let me give you an example, okay? Suppose there is an inflammatory focus. Why double-stranded formed RNA is not normal if this is formed? It is because AGP, during translation, ribosome expects a completely single-stranded mRNA. Only then ribosome will be able to read those mRNA's codons. Okay, so it will be able to translate only as long as it's a single-stranded RNA. But when siRNA comes and binds, it becomes a double-stranded RNA. So that is abnormal. Do you understand that? Yeah, this is interference. Yes, Dark Knight. We are discussing about interference. miRNA is microRNA, siRNA small interference RNA. Okay, so I'm going to tell you an example for how interference phenomenon can help you. Okay, suppose there is an inflammatory focus in a person and this inflammatory focus has switched on. No, Saisha, this has got nothing to do with CRISPR-Cas9. CRISPR-Cas9 is gene editing. Yeah, this is not CRISPR-Cas9. Watch this, okay? Yeah. So suppose there is an inflammatory focus in a person and this inflammatory focus has switched on multiple cytokine genes. So all these cytokine genes have undergone transcription and they've given rise to cytokine mRNAs. But suddenly something has happened, and or somebody has intervened and this inflammatory focus has been removed. In that case, will you want these mRNAs to be translated as cytokine proteins? If you allow these cytokine mRNAs to be translated as proteins, that will end up in unwanted hyperinflammatory state. Do you want such an unwanted hyperinflammatory state? No, this is what we call a cytokine storm. We don't want cytokine storm. So transcription is already over, but you don't want translation to happen. Do you understand this? Transcription is already over, but you don't want translation to happen. In that case, what are you going to do? In that case, you will allow all these siRNA genes to be transcribed. For every functional gene, including cytokine gene, for every functional gene, there are siRNA genes, and these siRNA genes are present within intronic sequences of those genes. So till some time back, we thought introns were non-coding. We thought introns do not have functions. But slowly we are picking up functions even for introns. And one of the functions that are exhibited by introns is that within intronic sequences, we have siRNA genes. So those siRNA genes will be transcribed to form siRNAs. And those siRNAs, let me show you siRNAs as blue color. So these siRNAs come and hybridize with those fully formed mRNAs. If you're asking me where will these siRNAs go and hybridize? Do they hybridize to the exonic sequences or do they hybridize to the untranslated region? This was also asked as an MCQ in INI CET. Can you see where I have drawn siRNAs? So this is mRNA with 5' 3' polarity. All your siRNAs hybridize to the 3' untranslated regions. Introns also have become functional, yes. Yeah, in school, not only in school, even in college, many times we teach introns or non-coding sequences, but sometimes introns also have functions, okay. So they go and bind where? These siRNAs go and bind to the 3' untranslated regions of mRNAs. Okay, so they go and hybridize here, and then when ribosome starts translating, let us denote ribosomes in black color. So when ribosomes start translating these mRNAs, they come across these double-stranded RNA structure. They don't like this. So what will they do? They recruit RNA transcription silencing complex, which comes with an endonuclease. It breaks all these mRNAs. Once mRNAs are fragmented, proteins will not be synthesized. So this is one of the ways by which your gene expression gets regulated. Because whenever you see about or whenever you say about regulation of gene expression, it's not just up regulation, down regulation is also regulation. Sometimes you want down regulation to happen, and that is done by siRNAs. So miRNAs or siRNAs take part in which phenomenon? They take part in interference phenomenon. And what do? They down regulate gene expression. They help in knockdown. Is that clear to all of you? It's a complex of proteins, pickle. Yeah, I will do molecular biology session. Okay. So miRNA is used for what? Will you get confused here anytime? They help in what? They help in gene knockdown. They down regulate gene expression. And knock out and knock in, this will be done by gene editing. Gene editing is done by CRISPR. Yeah, somebody was asking me about CRISPR, right? So CRISPR-Cas enzyme system, they bring about gene editing. Probably in the next YouTube session, I will have few questions on CRISPR-Cas. I'll help you understand about gene knockout and gene knock-in, that is gene editing. Okay. One short lecture on molecular biology. I'll do that AGP. I'll do it. Okay. Yes. So tell me what is the right answer here. miRNA binds where? This is This was asked in the same question paper, INICET 2021. miRNA binds to 3' untranslated regions. Actually, miRNAs bind anywhere, but most probably they won't bind to 3' untranslated region. Okay, so that's all for today. I will meet you again on 30th. Okay, so on 30th and 31st at 6 o'clock, I will have a session on molecular biology. We'll discuss PYQs, wherein most of the questions will be molecular biology based, so that you get a strong hold on it. Okay. Yes, AGP, you're right. Thanks, Pickle. So see you again. Good night.